Let's discuss transport of drugs across the cell membrane. Pharmacokinetic is derived from the two Greek words pharmakon, which means drug, kinesis, which means movement. It is a quantitative study of movement of drug in, through and out of the body. It deals with what the body does with the drug, which includes absorption, distribution, metabolism and excretion of a drug. Whenever a drug is given orally, it first reaches the GIT, gets absorbed from the GIT, then gets distributed throughout the body. Further gets metabolized mainly in liver and ultimately gets excreted from the body. All this pharmacokinetic process involves transport of drug across the biological membranes. All biological membranes are made up of lipid bilayer, mainly phospholipid, which forms flexible and dynamic barrier. The dynamic nature of the lipid bilayer is crucial from the membrane's function, including cell signaling, transport, and maintaining the structural integrity of the cell. Drugs cross various biological membranes by the following mechanisms, such as passive diffusion, facilitated diffusion, active transport, endocytosis or exocytosis, and filtration. In passive diffusion, drug molecules move from the region of higher concentration to the lower concentration, that is, along the concentration gradient, until the equilibrium is attained. Since the movement is along the concentration gradient, it does not require energy. Therefore, the rate of diffusion is directly proportional to the concentration gradient across the membrane. It is the process by which majority of drugs are absorbed, mainly lipid-soluble drugs, gets transported across the cell membrane by passive diffusion. Passive diffusion does not require carrier for the transport, while facilitated diffusion is carrier-mediated transport of a substance which is also along the concentration gradient. Thus, it does not require energy. Here, the drug attaches the membrane and facilitates its diffusion across the membrane. For example, transport of glucose across the muscle cell membrane by GLUT4 transporter. Next is active transport. Here, the drug molecules move from the region of lower concentration to the region of higher concentration, that is against the concentration gradient, thus it requires energy. For example, transport of sympathomimetic amines in the neural tissue, transport of choline into cholinergic neurons, and absorption of levodopa from the intestine are the examples of active transport. The active transport are further classified as primary active transport and secondary active transport. In primary active transport, the energy is obtained by hydrolysis of ATP. For example, sodium-potassium pump. This pump uses ATP to move sodium in the cell and potassium out of the cell. In secondary active transport, energy is derived from the transport of another substance in the same direction. For example, sodium glucose cotransporter, which moves glucose into the cell along the sodium ions or in the opposite direction, that is antiporter. For example, sodium calcium exchanger, which moves calcium ions out of the cell while sodium ions into the cell. Next is endocytosis or exocytosis. Certain substances which are very large or impermeable molecules are transported inside the cell by endocytosis or excreted out by exocytosis by enclosing their particles into tiny vesicles. Endocytosis can further be classified as phagocytosis, that is cellular eating, and pinocytosis, that is cellular drinking. This depends on the nature of material being engulfed. For example, vitamin B12, which is an intrinsic factor complex, is absorbed from the gut by endocytosis. Coming to the filtration, Filtration depends on the molecular size and the weight of the drug. If the drug molecules are smaller than the pores, they are filtered easily through the membrane. Thank you.